Okay, hi guys. So it turned out yesterday the microphone had stopped working somehow, and so I apologize. So I'm going to remake this recording and post it for the second half of chapter 9. So today what we're going to be talking about are mobile genetic elements, viruses, and then examining the human genome. So in this Albert's book, it's the second half of chapter 9. Last time, if you look at the part, you will see that it talked about, we talked about genetic variation generation. So how do we introduce new variations in our genome, in an organism's genome, and what kind of ramifications it has, how does it affect the organism itself, how does it help them get more variability, and then how does it change the species over time to evolve into other things. And then using this information, we can reconstruct how different organisms and different species are related to each other, and thus construct a family tree of sorts for all different organisms present in life. So today we are going to be looking at another way that variations as well as changes happen in our genome, in an organism's genome, with the help of mobile genetic elements or jumping genes and viruses, which can also act something similar to these mobile genetic elements. And then finally we'll talk about how we can examine the human genome for these changes, and then look at them to identify an individual within a population, or to compare different populations and see which one derived from where, how things migrated from one place to another. So let's get started. So the first part of this is going to be about mobile genetic elements and viruses. So mobile genetic elements, also many of the are termed transposons, are jumping genes. All of those are basically elements. They are sequences of DNA that contain information within it that allows it to be moved from one place to another. So these sequences will contain essentially sequences for genes that can allow it to be excised or cut and then moved to another place and pasted in a different part of the genome, adjacent to it, or at another place, homologous to it, or another place entirely. It can also have sequences that allow it to be replicated and made more copies of, which can then go and insert themselves elsewhere in the genome. So these are basically two different mechanisms with which they can move. The most common mobile genetic elements in bacteria are usually DNA-only transposons, so they are DNA sequences. And that's what you're working with. So here is an example where you have a donor DNA, essentially a sequence of DNA that has the transposable element, the transposon, the mobile genetic element in it. If this was inserted into the host bacteria, this transposon will go into the target DNA sequence due to the existence of sometimes these sequences that are homologous to the donor, the target sequence, that allow it to kind of combine in there. They are cut. Transposons themselves are usually cut and paste transpositions, so they are cut out, excised out of the donor DNA, and they then insert themselves back into the adjacent DNA or the target DNA, leading to the target DNA having the new DNA sequence, and your donor DNA not having that sequence anymore. So it loses the transposon in the donor DNA and gains it in the new DNA sequence. This is called cut and paste transposition. The other type is what is called replicative transposition. In a replicative transposition, the transposon in the donor DNA remains there. It does not get excised and put into the donor DNA. It is instead copied into another sequence, so that there's another copy is made of that sequence, and that double-stranded copy is then inserted into the donor DNA, the target DNA at the end of the day. So you end up with two copies of transposon in that genome at that point. So that's the basic difference. In the first one, the number of transposon copies that are there remain the same. It's still 1, but in replicative transposition, these can be replicated. So you can imagine that now once there are two copies, both of them can copy themselves and then go and insert another location as well. And then you'll have 4 of these sequences. And this process can go on, multiple times depending on what type of transposon it is, what makes it copy and paste, and how often it happens. So that can obviously really lead to changes in the final output. So looking at what exactly is needed for this process to occur, the basic need for a transposition is a gene called a transposase gene. So the transposable element, the mobile genetic element, contains this gene. That's what makes it a transposon. In addition to that, they can also contain other genes that can help it do other functions. And sometimes it can contain genes that are not responsible for anything related to transposition of that transposable element, but are actually impart other functions to the organism in which they end up in. So for example, here is an example, this TN3 transposable element, the mobile genetic element. It has your transposase gene, obviously, because that's needed, and then it has an ampicillin resistance gene. So if this particular sequence was to be input into a bacteria, that particular bacteria would end up becoming ampicillin resistant, so that if it was given or grown in the presence of ampicillin, it would be able to grow. As opposed to a bacteria that does not have that, it would die and would not be able to grow in the presence of ampicillin. So this is actually how you can create antibiotic resistance, and the spread of these transposable elements in different organisms, if they were able to move from one bacteria to another, which they are in many cases, and leads to the spread of these, how much they spread, will develop more and more antibiotic resistant strains or bacteria in the world. So that is an important consideration for these transposons. So now looking at what can they do. Once you have a mobile genetic element in a genome, it can lead to many different outcomes. Right, so many times these mobile genetic elements insert themselves into intronic sequences. So if they're just hanging out in the intron, that's not going to be that big of a problem as far as the actual gene product is concerned, because most genes themselves, the gene product that you're looking at, the protein, is going to be translated from the exon sequences, the sequences present in the exon sequence. However, these introns can contain regulatory genomes or regulatory sequences in them, and those can be interfered by the insertion of these mobile genetic elements. They can also cause another problem if there is a transposon that is found multiple times in a given sequence for a given gene. So for example, this particular gene, example gene sequence, gene A, contains 2 mobile genetic elements that are very similar. So these are 2 very, very similar transposable elements. They are both in introns, so as such, they don't really affect gene A. Gene A is still going to produce the same protein it always did. However, because they are very similar, their ends can be misrecognized by the transposase so that it can accidentally cut out or excise when it was doing that cut and paste transposition, as we talked about. It can excise these 2 sequences in a way that some of this mobile genetic element remains in the original gene A, while you end up with this fragment that contains enough sequence for it to still be a transposable, to still be pasted into the new place, but it loses a lot of this mobile genetic element, right? A lot of that information for it to remain a transposable element itself. However, now you have this little fragment. It was cut out, excised out by the transposase, and now you have this sequence that's going to be inserted somewhere. Now, this can insert itself two different ways. It can insert itself into gene A the way that it did before, or into another intronic sequence as it did. But now there's a problem. This fragment is not just transposable element. It has a fragment of DNA that contains introns, but also contains one of the exons from that original gene A. So that original gene A now is disrupted, because when it now becomes a transcript, it will be missing a big chunk of it, and it's going to alter the protein that comes out of it. Maybe that no protein will be made, or if it is made, maybe it will be truncated or be missing a large domain. And this particular fragment, when it gets inserted somewhere, let's say it gets inserted into another gene, now that particular gene has suddenly gained an additional sequence, an additional exon that can now either render the whole protein useless because of the way it changes the sequence, or it may end up with a new protein that wasn't there before, that has new domains. Maybe it contained a DNA binding domain that didn't exist before, or a nuclear localization signal that didn't exist before, and now this protein is able to translocate to nucleus when it was supposed to do function in the cytoplasm or outside. So that's going to cause obviously a disruption of or alteration of that protein's structure and function. So what mobile genetic elements moving in this way can lead to generating obviously novel genes and new proteins and new functions, but can also alter gene regulation if the same thing happened where these mobile genetic elements ended up in a regulatory sequence of the genome. So now looking at it in humans, humans contain also similar to bacteria. They have two different classes of transposable sequences. Two major families, class 1 and class 2. They essentially work very similar to cut and paste versus replicative transposition. So class 1 is actually similar to what you would call a replicative transposition. So this is a retrotransposon. This transposon does not just cut and paste. This is kind of like that one that will replicate and then give another copy. So now you will have two copies instead of one at the end. So in this case, this will transcribe the sequence that's present there into an RNA intermediate, which is then changed into DNA intermediate through reverse transcription. And once that double-stranded intermediate is made, it will get integrated into the target DNA. So in this case, you're going to keep that donor DNA with the retrotransposon in it. You're going to transcribe it into RNA intermediate, then reverse transcribe it back into DNA, and then get integrated into a new sequence. So you end up with two at the end of the day. On the other hand, your DNA transposons, which are class 2, are ones where transposase is present within that sequence. It cuts it. It has then that fragment of DNA that is excised that will then get integrated into the target DNA to lead to essentially transposon jumping into a new place. So transposable element, a mobile genetic element in that way. So looking at a retrotransposon, because it is using an RNA intermediate, it's going to need additional supplies, right? It needs a way to be able to transcribe that DNA and then reverse transcribe it into a double-stranded DNA copy. However, it then has this extra ability to create multiple copies of itself and put them all over the genome wherever it gets inserted. So you will end up multiplying it, and you'll be able to see it many, many, many times within the genome. And obviously that's going to increase the size of the genome, but also increase the variability in it, because one person may have 100 copies of this particular retrotransposon while another person may have 500, depending on how many times it was copied and pasted. So that's how essentially we look at mobile genetic elements being a driving force for evolution. because they can introduce variability within the genome. They can introduce new sequences, they can disrupt genes, they can add different domains to the genes when they're excised and pasted improperly. They can introduce different regulatory stuff by inserting themselves into regulatory portions of the genome. So a lot of things that they can do. Now, viruses can act very similar to transposons themselves. They come in many different shapes and sizes, and they come in two different flavors as well. You have DNA viruses that have double-stranded DNA inside them, or DNA molecules inside them. So pox virus, herpes, and adenoviruses and papillomaviruses are all examples of that. Now, monkeypox, for example, the new virus that is floating around, scaring us to be the new thing, new scary thing, and that's a double-stranded DNA virus, right? So because it is a double-stranded DNA virus, it is rather heavy, which is why even though which is why it's a contact virus. It's not just going to be, you can't just get infected with aerosol particles because it's too heavy because of the double-stranded DNA. It would just kind of sink down. So while it can survive in the air for a little bit, it's not going to be very easily transferred through the air, which is different than the coronavirus, obviously. With the COVID, the issue was that you could have that big population of these viral particles in the air suspended, and you could get it with close contact. With this monkey virus, you should be able to be a little bit more protected. However, it is still obviously contagious in some ways. So you still want to be very careful with the close contact stuff. The other types of viruses are the RNA viruses, and that's similar to the coronavirus, the COVID, for example, and a lot of these other viruses that have RNA. So some of these viruses can actually serve as transposons themselves because they can do the same thing. They can get their genome can get integrated into human genome or the organism genome that they infect, and then they can hang out there and make more viruses, and then they can also get excised out at different times and then get transferred to other places. So let's see how that would work. And we'll talk about it a little bit by examining both DNA and RNA viruses. So first of all, here's just kind of a list of some viruses, common viruses that we may see, whether they're double-stranded DNA or single-stranded RNA viruses, and what kind of diseases that may cause. And now we're going to look at first just like general biology of viruses. So viruses cannot just replicate all by themselves. They need a host. Whether they are DNA virus or an RNA virus, they need to infect a cell first. And so many times they will infect a cell by binding to the cell membranes, and then they will inject their genome inside, leaving the viral coat behind on the cell membrane. This DNA, in this case, this is a DNA virus. Once that genome of that virus is injected, in this case, since it was a DNA, it could just be replicated just like normal, right? So the cell is going to replicate that DNA and make more copies using the cell's own replicative machinery. And then once it is transcribed into RNAs, those RNAs are going to be translated using the ribosomes from the infected cell. And then those viruses, viral mRNAs, when they are translated, will be made into those proteins that are components of the virus. Once those proteins are made, they do self-assembly into viral particles using the genome and the copies of the genome from the DNA, along with all these different proteins, and then they will exit from the cell, usually through lysogeny, which means that the cell will rupture, and these particles will just kind of spread to neighboring cells, infect those, and the cycle will continue on. So that's going to be, in this case, a very clear-cut process, where it's very little chance of this particular genome integrating into our genome and then causing any issues. However, that's not always the case. It can happen. And the other thing that can happen is where you have an RNA virus. Now, if you have a retrovirus, or a virus that's made of a single-stranded RNA, they will still come in again to the cell using similar mechanisms as before. However, in this case, they actually don't come in with just the genome. What they come in with is the genome and the reverse transcriptase that are in the actual viral particle. And when they enter into the host genome or host cell, when they infect a cell, they only lose the outer envelope, and there's still a protein coat that is protecting that RNA genome and the reverse transcriptase that's already bound to it. Once the RNA, this protein coat contained RNA has entered the cell, it is then going to release the genome, and that genome, when released, will be transcribed, right? Back into, so using that reverse transcriptase, it's going to be transcribed back into the DNA. That's why it's called a retrovirus, because it's going in the opposite direction. So now you have this RNA-DNA hybrid that obviously is not going to be very stable, so it's going to be retranslated to make a DNA, double-stranded DNA molecule. This double-stranded DNA molecule then gets integrated into the host cell, in the host genome, whatever genome it is, whether it is mouse, rabbit, bat, humans, whatever type of organism that it is infecting, it's going to integrate into that host genome. Now, it can hang out there for a while, or it could just straight away go into infective mode, in which case it's going to transcribe many copies. It's just going to use the transcription machinery again of the organism itself, of the host cell, and it's going to make many RNA copies. Those RNA copies will contain, obviously, all the different parts of the virus, all those genes related to the viral proteins. Those proteins are going to be translated. You're going to get all the main components, all the building blocks of the virus that are then going to be self-assembled again spontaneously, just like before, into new infectious particles, which will then be released into the environment, ready to infect other cells. So in this case, infection by retrovirus includes a transcription and integration of viral genome into the host cell DNA, which was missing when we were looking at just a double-strand DNA virus. So in this case, because it's integrated into the viral genome, if it had just hung out there, yes, it's disrupted some sequence in there, and it maybe disrupted a regulatory sequence or a gene sequence. Still, it's not just affecting that cell. It can also at another time be excised back out in some cases, in which case, if it is excised out improperly, it can take part of the host genome with it to the next cell, where it can cause recombination to occur and changes to occur. So there are multiple ways, again, that viruses can also act very similar to mobile genetic elements and act as driving force of variability introduction or variation, mutations, and evolution. So just to kind of recap, mobile genetic elements must have transposons, that is, must have some elements inside them, some components that they need to be able to move from one place to another. This is in the form of the transposase gene. The human genome contains two different types of transposable sequences, class 1 and 2. Class 1 are the retrotransposons, where you have an RNA intermediate, and class 2 are those sequences where it is just the DNA itself that is the transposable sequence. Viruses can also act very similar to mobile genetic elements, but they can move between different cells and organisms, right? Different organisms too sometimes. Retroviruses are the ones that specifically act very similar to those retrotransposons and reverse the normal flow of genetic information, where you have an RNA transcript that gets transcribed back into a DNA, integrated into genome, where it starts the cycle back again. Now, looking at how we can utilize all this information to examine our human genome, let's kind of think about our genome for a second and just appreciate how amazing and big and large it is, right? So there are 3 billion nucleotide pairs in the human genome. So obviously that's a lot of information that's put all inside it. If you were to take that genome and just kind of stretch it out in a linear manner, that particular DNA could essentially cover this entire space that is put at this red line. That's how long it would be. You could bisect that African continent at that point. At that scale, and that's a really large area, there would be a protein coding gene, an actual protein coding gene every 150 meters. So there would be a lot of genes. In any one of these genes, obviously genes have a very large variability in the actual size, but they are on average about 30 meters in length. Those 30 meters contain both exonic as well as intronic sequences. So if you were just to excise out all the introns and just combine the exons together, that gene would be only 1 meter long. So starting from 30, that's a really tiny amount. So majority of that sequence at the end of the day is those intronic sequences. The rest, would be all introns. So looking at this visually in another manner, if we were to look at one of the human chromosomes, in this case this is chromosome 22, you can look at it in a chromosome form, right, when it's fully condensed in that heterochromatin state. And it has a short arm and a long arm. And here you have the two sister chromatids combined, stuck together at the centromere. So if you were to magnify a portion of it and look at just a 10% portion from this particular chromosome, just 10% of chromosome 22 in the long arm, that region contains about 40 genes. That's a whole lot of genes. And if I were to look at, again, a small portion of it, I see 4 genes in this sequence, right, in this 1% of the chromosome. And within there, if I were to look at a particular gene right here, you notice that in this single gene, there are, first of all, 340,000 nucleotide pairs, out of which majority of them are just introns, and you have these tiny exons that are going to be spliced together to create your final protein product. So that's, again, a whole lot of extra information that we don't yet understand completely what that function is. Some of it is regulation, some may have other functions, but it is more than what we know at that time. So this, again, gives you some statistics about how many, out of those 3 billion nucleotide pairs, how much of that actually contains useful, what we consider quote-unquote useful information. Those 3 billion nucleotide pairs contain about 19,000 genes. They also contain 5,000 other genes that are non-protein coding, so they may be coding for microRNA or ribosomal RNA or other sequences that are important. And then the remaining portion contains some regulatory sequence and some highly repetitive, high copy repetitive number elements. The mobile elements, these mobile genetic elements that we've talked about, pseudogenes, that are genes that may have existed at some time but have now become dead because they have become truncated or something's happened to them so that they are no longer able to be transcribed or translated appropriately. So the bulk of your genome essentially is made of these repetitive sequences, whether they are tiny small ones or larger ones, and contains non-coding DNA. And very small percentage, if you look at this percentages, only about like 3 to 4% is protein coding exons. Everything else is either introns or non-repetitive DNA that may have some regulatory function, or that's not part of the protein coding sequence for sure. And then simple repeats, transposon, retrotransposon, signs, and lines. So there are a lot of different types of repetitive sequences that are present there. Some that are very small, just a few nucleotides long, and some that are larger transposon and larger elements. And so comparing different genomes, examining human genome against a lot, some of the other species, some of the other organisms that are out there, we actually find that human genome is not entirely unique, but is a little bit different than the other genomes in its complexity and its amount of useless information. It's not useless, again, but in the amount of non-protein coding information. So while other genomes, other eukaryotic genomes like of yeast, contain majority of that sequence is protein coding, gene coding sequence. In humans, only about 3% of it is actual gene coding sequence, while the remaining stuff is not. So that makes our human genome very less, a lot less dense, gene dense than these other organisms. Now that could be an advantage in the sense that because it has so much of this extra stuff, when things go wrong, when mutations happen, they're more likely to happen in those areas that are there, and less likely to happen in those small important sequences. So it can be a way of protection too, in a way. But there is a lot of this information there. So because it is so complex, one of the problems that arises from it, and even without it, if we were looking at, let's say, even a 10,000 base pair genome, it's hard to identify protein coding genes, right? Because you have to consider that they can be, considering the length of the codon, which is the 3 nucleotide sequence, they could be in any one of the 3 reading frames, and looking at all the possibilities, it would be really hard to find initiation codons and stop codons and possible protein coding sequences. So, and especially if you take into account then the introns and exons. So now, with the help of computer programs, we can easily do that no matter how large the sequence and how complicated the genome. So for example, human genome with all these extra information and all these repetitive sequences and all the introns and exons can become extremely complicated to decipher. But using computer programs, you can read all 3 possible reading frames of each of your DNA strands, starting at any given position all along the genome. And you can identify any possible, in this case, all your reds that you see are your possible initiation codons in which of the 2 strands of the DNA they are on. And then it can also identify your stop codons and look at possibility of you having those various open reading frames or protein coding genes. So that's a big application of computer programs that we use all the time as we sequence more and more genomes. Another thing we can do, another application that we can have with computer programs to examine or characterize these protein coding genes. So one is to identify possible protein coding genes. Then the second is to examine and characterize these genes and to look at presence of possible exons and intron sequences in there. And again, some of it can be done using the sequence information. Another thing you can do is you can extract, and this is an example from the beta of a portion of the beta-actin gene that contains several exons and introns in it. You can extract the RNA from these various cell types, and you can sequence them and look at how many copies of each of these regions are present there. And what you will notice in this case, if you were to look at a particular given sequence, and you can look at the number of copies that are found, or number of reads, which is essentially number of copies of a particular region that is found, you will notice that your exons are in a higher number of reads than the intronic sequences. So you can kind of guesstimate where your introns would be and where your exons would be. And this is probably because these exons are going to be part of the mature RNA, while the introns are going to be still there. They're going to be in that original transcript, but then they're going to be spliced out, so they'll be gone. So that's why you still get some mRNA reads or some RNA reads from those intronic sequences as well. And finally, another thing that we can do with these sequence information, examining the human genome, is using, again, information from genome sequences. We can trace the movement of a particular species, of a particular organism from one place to another, and we can look at how long they took or how variable they got over that time. So here in this example, we are looking at descendants of modern humans, or just development of modern humans from Neanderthals, and looking at how they moved from the central region into various parts of the world, and how divergent they got in their sequence as they moved to a later place. So this type of scenario would include examining the genomes from fossil records, and obviously, genome sequences from the Neanderthal population, and then examining the fossil records and looking at extraction of DNA from some of these and from people in different areas, and comparing them to the Neanderthal genome sequences that we are able to establish from those studies. So, oh, sorry, why did it go there? So in this case, remember that we had humans that were leaving Africa around 130,000 years ago, and then they were kind of settled down and hanging out in this region where there was a lot of interbreeding within that population. And then as they moved to various locations across the globe, they diverged more and more, which can be seen in the fossil record as well as the genomes from humans in various places. They can all be traced back to that Neanderthal population that was inhabited about 55,000 years ago. Lastly, we can use, we can, like we said, we use genome sequencing information, genome sequencing to identify this movement and to identify when things are related. We can also use similar type of genome sequencing to identify different individuals from another, and which is used in forensics as well, to identify a particular sample to be from a particular person, or to exclude somebody from the suspect list, for example, due to absence of certain genome sequences within them. Whenever we are comparing two populations or one individual to another, what we are usually using are those repetitive sequence areas, or those areas with high levels of variability. So one of those things is what we call single nucleotide polymorphisms, or SNPs. The single nucleotide polymorphisms are small, one single nucleotide changes in specific portion of gene sequences or of genome that can be present within a particular population. So they're just different. Those are specific points in genomes that are just different by a single nucleotide pair, while everything else surrounding it is still contained the same. So here, this is approximately 1,000 nucleotide pairs that are being shown, and you can see these in this region, and these are these two SNPs that are present within that region that can be different in different groups, different populations. And so if we were comparing DNA sequences from 4 different individuals from 4 different places, you could look at these SNPs in these locations to locate whether they were coming from the same population or whether they were derived from the same population or not. So one thing to remember about SNPs is that to be classified as a single nucleotide polymorphism, it has to be a single nucleotide change in the sequence that does not affect the final outcome of the protein. So it's still going to lead to the same protein sequence. It's not going to have any effect on it. All these sequences would still lead to the same product, even though the nucleotide is different. Again, due to the wobble hypothesis, you can have that last nucleotide change and still give you the same amino acid, for example. In general, there are about 1 SNP every 1,000 nucleotide pair. And so you can look for those, and then you can use those to identify different populations and different individuals. Typically, you can classify a SNP in a particular population if at least 1% of that population has that particular polymorphism. So going back to this, the nucleotide sequences, just kind of reviewing this, that all the nucleotide sequences in human genome can show us how different genes are arranged. If we were to, examining them, we can find protein coding sequences versus non-protein coding sequences and look for differences between them. If sometimes you have 2 organisms with a pretty much same exact genome with a very little difference, but it's the gene regulation that is different, that is disrupted or that is altered, that leads to a completely different outcome in the type of organism that you're looking at, and how they're different. If we were to examine the genome of extinct Neanderthals, we can look at how much of that is different from us as modern day humans. Given that the genome is very similar, there are definitely variabilities and variations that have led to the existence of modern day humans. And finally, the genome variations that's there helps us identify individuals and populations and their connection with each other. So hopefully this was useful, and hopefully this time the volume and voice was good. Next time I'm going to be uploading chapter 10. And I'll try to upload it within the next couple of days, but it may be next week, depending on when I'm able to find the time. So until then, take care. Let me know if you have any questions.